Hello, and welcome everyone. In this podcast, we're going to begin by reviewing some terms that are important in understanding cell division, and then we're going to talk more specifically about cell division. Following this podcast, we'll have a podcast on mitosis and one on meiosis and gamete formation. So let's review some basic terms we need to know as we move forward. We should know the difference between a somatic cell and a gamete. Let's start with gametes here. Gametes are the sex cells. So we can think of gametes as being sperm or oocytes. In organisms like fungi, which are also eukaryotic, of course, they don't produce sperm or eggs, but they do produce spores. We can also think of gametes as in terms of their ploidy. How many chromosomes do they have? Gametes are haploid. Or we might call them 1N. Somatic cells are all cells except gametes. They are diploid, and we often call that 2N. Humans, for instance, so let's write humans here, we have 46 chromosomes in our somatic cells. In our haploid cells, in the sperm or eggs, we have 23 chromosomes. But you should be able to answer this for any organism if I tell you one or the other. It's always half the number. So if I tell you there is an organism and they have 52 chromosomes in their somatic cells, how many would be in their gametes? You should be able to tell me that there are 26 chromosomes in their gametes. Somatic cells are produced by mitosis. And we'll talk about mitosis here in just a moment. Well, rather on the next podcast. Gametes are produced through the cell cycle, cell division, known as meiosis. I think that's enough of a comparison between these two. And we've had this before, but it bears just repeating again just to remind ourselves. When we look at chromosomes, let's just draw, say in humans, here's chromosome 1, and here's chromosome 22. I picked these two because they are noticeably different in size. We would call these two homologous chromosomes. And we'd call these two chromosomes homologous chromosomes. We would say that chromosome 1 here is non-homologous with chromosome 22 here. So in this picture here, we could say we have two homologous chromosomes. In editing this podcast, I realized I should have said two pairs of homologous chromosomes and not just two homologous chromosomes. The better way to say it is two pairs of homologous chromosomes, just so you have it right in your notes. We could also say we have 4 chromatids, and we'll define chromatids a little bit later when we talk about cell division and mitosis, but essentially a chromatid is one of these. So this is one chromatid, this is one chromatid, this is one chromatid, and this is one chromatid. The way I have drawn these are both unreplicated. If they were replicated, I would have drawn another chromosome attached to it, like so. The way I've just drawn this with the dashed line here, now they would be replicated chromosomes. I'm going to go ahead and erase this dashed chromosome for now, though. And as a reminder, these chromosomes, each of these chromosomes have one long piece of DNA on them, and they have genes associated with them, of course. So we may have a gene here, a gene here, a gene here, a gene here. Same over here. And really, there would be thousands of genes on the chromosome 1 and several hundred on chromosome 22 here. I just drew a few. And these are homologous chromosomes, so they're going to have the same genes on the other sides of them. Remember, homologous chromosomes are identical in the sense that they're the same length and they have the same types of genes all the way down. Now, it's possible that they could be different alleles. So this could be big A, this could be little a, this could be big B, this could also be big B, this could be little c and big C here, little d, little d. So these are just different kinds of alleles. So big A, little a are two alleles of the gene A. Big B are the same allele of the gene B. Little c, big C are different alleles of gene C. Little d are the same alleles of gene D. Now they still function in the same pathway. Let's look at one up here, big A, little a. They still function in the same pathway, but with having a different allele, it may lead to a different phenotype. We would say, in this case, B would be an example of let's write that over here. B would be homozygous dominant. Little c, big C would be heterozygous, as would be big A, little a. And then little d would be homozygous recessive. Sorry, I ran out of room here. Another thing we should point out is the term locus. Locus is just the physical space that the gene resides. So the locus for gene A is right here on this chromosome 1, on both of the homologous pairs. So that would be the locus for gene A, this position on the chromosome. The locus for gene B would be this position on the chromosome, and so forth as you move down through gene D. It's just the physical location of that gene. Okay, now with some of these basic terms down, let's go ahead and talk about cell division. And before we get started with what eukaryotes do, let's talk about the way bacteria divide. And they divide asexually. And they do so by using a process called binary fission. And that's the main thing I want you to know, is that they don't undergo the same kind of mitotic and meiotic cell divisions that we see in eukaryotes, but they use a specialized one called asexual reproduction or binary fission. But you may have your E. coli cell here, and you have the DNA here, the circular chromosomes here. They will have their DNA undergo replication just like we learned about before. You will double the size of the chromosomes, so you end up with 2 copies. And then the cell begins to divide. And in the division process, the chromosomes that were just copied here end up in either side of the cell. And then eventually they'll complete their division, and you'll end up with a copy of the chromosome in each one. And that's all we need to know about binary fission for now, or for this class anyhow. Before we move away from binary fission, let's go ahead and define a couple of quick terms that are going to be relevant as we move forward with cell division. And that is, what do we call this original cell? We call this original cell the mother cell. And then the resulting cell here we call a daughter cell. Same over here, we call this one a daughter cell too. And when we talk about eukaryotic cell division, we'll use these same kinds of terminology. This same kind of terminology. Okay, let's go ahead and talk about eukaryotic cell division. So we can think of eukaryotic cell division in 2 different branches. We can think about mitosis And then we can think about meiosis. During mitosis, you have one mother cell, and let's make it a diploid cell. In some cases, it doesn't have to be diploid, but we'll make it diploid, 2N, and it divides through mitosis, and it generates two genetically identical cells. This is important because this is going to occur in, say, multicellular organisms' somatic cells. Because when you have a cell divide in your lungs, you want to make sure that that new cell, the new daughter cell, is also a lung cell, so it can perform the same function. So the key here is they are identical. So they're still going to be 2N diploid. Now meiosis, we'll see there are some similar features between mitosis and meiosis, but one of the big differences is that meiosis is divided up into two separate divisions. The first is meiosis 1, and the second is meiosis 2. And we're going to go through both of these in excruciating detail. But by the end of meiosis 2, we end up with four cells. And if we start off with a diploid, each of these cells will be 1N. So you go from a diploid cell, a single diploid cell, to four haploid cells. And they are genetically different from the parent cell. Not only do they have half the number of chromosomes, but through meiosis 1 and 2, particularly meiosis 1, various mutations can be generated into these different haploid gametes that are the product. So we have different cells come out. In the next podcast, we're going to focus on mitosis, and then the following one we'll focus on meiosis. But for the rest of this podcast, I want to talk about the cell cycle. What this cell does to prepare for mitosis or to prepare for meiosis. So let's go ahead and draw our cell cycle here. Here we have G1, and then we have S, then we have G2, and then we'll have the M phase, which could be mitosis or meiosis. So let's first talk about what happens at each of these stages, and then we're going to talk about the actual cells, or rather the chromosomes within the cells, and see what's happening with them. So during G1, we call this gap 1. It's the gap before DNA synthesis occurs. During G1, the cell is preparing to copy its DNA. It needs to make sure that the DNA doesn't have mutations, so it checks for mutations. And it makes sure its enzymes are ready to enter into this next phase. And S phase stands for synthesis. This is where DNA replication occurs. And DNA is actively copied during the S phase. At the end of the S phase, it enters into G2. In G2, we call this gap 2. Cell growth occurs here, and it essentially prepares for M phase, mitosis or meiosis. It makes sure enzymes are necessary. It also makes sure that all the DNA has been copied and there are not mutations, and prepares the nucleus for division. During M phase, the new chromosomes that were just copied here in S phase, so chromosomes, separate. Now this happens in mitosis or meiosis, but there are some differences that we'll certainly focus on when we get to that point. Okay, let's erase this now, because now we want to focus on the chromosomes and what's happening to the chromosomes as we go through the cell cycle. And for now, I'm just going to show one chromosome as we go through the cell cycle here. So here's the cell, and I'm going to draw one chromosome here. This is a homologous pair of chromosomes. If this were, say, a human cell, there would still be 22 other pairs of chromosomes, but we're just going to show one here as we move around here. As this cell leaves G1 and enters S, the DNA will be copied in S, so we're going to copy our chromosomes. And I'm going to put the copied one in a different color. Then, as you move into G2 phase, the chromosomes will appear the same as they did in S phase. Then as they enter into M phase, during M phase, and we're going to focus on mitosis for now, these chromosomes will be pulled apart from each other, and the cytoplasm will divide to produce two cells, and you will end up with two homologous chromosomes in each one. Now these cells can enter into G1 and divide again, or they can enter into a subset of this that we didn't really talk too much about, but a cell, instead of going through division, may kind of check out of the cell cycle for a little bit and enter what we call G0. In G0, this cell, depending on the cell, may be temporarily out of the cell cycle, or it may be permanently out of the cell cycle, just depending on what kind of cell it is. If it's a neuron, it's permanently out of the cell cycle, but if it's your skin cell, it's not permanently out of the cell cycle. It will come back into the cell cycle, just depending. All right, I want to go over a couple terms here and count how many chromosomes we have at each stage. I want to count how many. I want to also talk about how many chromatids we have at each stage, and finally how many sister chromatids. And I'll use I'll place these numbers using the different colors, brown, orange, or purple, so you know which ones I'm talking about. So in this stage here, we have 2 chromosomes. You could also say you have 1 pair of chromosomes, but we have 2 chromosomes here. When it's in G0, it's 2 chromosomes. In S phase, it's 2 chromosomes. Even though we see 4 copies here, it's still 2 chromosomes, because as long as they're attached at the centromere, we still call it the same chromosome. Over here we have 2 chromosomes. Over here we have 2 in each of these new daughter cells. Now, how many chromatids do we have? Well, each one of these is a chromatid. So here we have 2 chromatids. Here we have 2. Here, however, we have a new number, 1, 2, 3, 4. Even though it's 2 chromosomes, we say it's 4 chromatids. Here is 4, and then when they divide, once again, we go back to 2 chromatids. Now, what about sister chromatids? Well, here we're going to say 0, and I'll explain why in a moment. Here we're going to say 0. Here we're going to say that these 2 are sister chromatids. I'm going to put SC for sister chromatids. And these are SCs. So it's not really saying how many there are. It's just recognizing that after the DNA has divided, that copy of that chromatid that is attached to the original one here, we call these sister chromatids. So they're still chromatids, just like we counted before, but we say they're sisters because they are identical. And same thing here. These are each sister chromatids as well. So as long as there was no mutations made during DNA replication, these 2 sister chromatids should be identical. A common mistake that is often made is getting sister chromatids confused with homologous chromosomes. So I want to make sure I kind of go over that again real quick. We call this a homologous chromosome with this one. These are both, say, chromosome 1 in human. They have the same order of genes, they're the same length, but they're not necessarily identical, right? Because you could have a big A allele here or a little A allele here. So they're not identical, but it's the same genes in the same order. Sister chromatids is when you take these homologous chromosomes here and you copy each one of them. And when you copy each one, each half of the homologs here are identical. So over here, I don't have a lot of room to write this, but I'll try. This over here was big A. This here will also be big A because they're identical, because they just copied each other. Here is little a, and over here is, I'm going to write it up here, it's hard to read it, but it would also be little a, because these two are sister chromatids. They're identical to each other. Alright, that ends this podcast. Let's end with a short summary. So in this podcast, we talked about some basic terms. And you should be familiar with the differences between somatic cells and gametes, how many chromosomes are in each one, the kind of cell division that occurs to create each one of them, and where you would find them in the body. You should know the difference between diploids and haploids. Know that diploids contain twice the chromosomal content than a haploid cell. Homologous and non-homologous chromosomes, you should know the difference between those two when you're looking at two chromosomes. Or if I bring back a picture of a karyotype, you should be able to identify homologous and non-homologous chromosomes there. Alleles, genes, and locuses, or loci I should say in plural, you should know what they are, where they're at, and how to identify them. You should also know the difference between homologous and non-homologous. We then talked about the cell cycle, or rather I should say cell division, because we talked about what happens with bacteria, how they undergo binary fission, and then we focused more on the eukaryotes here. You should be able to draw the cell cycle with the different parts, G1, S, G2, and the M phase. You should know what happens at each of these phases in general term. What's the cell doing? What's happening inside the cell? You should know what's happening to the chromosomes as you go through this. When do they get copied? We spent quite a bit of time talking about the differences between chromosomes and counting the number of chromosomes here, the number of chromatids, and the number of sister chromatids, and what's different between a sister chromatid and a homologous chromosome. That's all I have for this podcast. If you have any questions at all, please let me know. I'll see you in class. Bye.